Bill. So continuing with the subjects from yesterday, let's delve more into how genomics can be informative about regulatory regions. Now, I had sort of this cluster of additional things that we could talk about, and of course, we won't get through it all, right? Because I want you to ask questions and we'll have discussion. And even though it's near and dear to my heart, I'm going to skip over the conservation part. This has to do with if you see a feature in mouse, how likely is it that you'll see it in an orthologous location in human, and what does that mean is very important. But actually, based on some feedback I've been getting questions, I think we ought to get into the different assays for enhancers, what they mean, and issues with interpreting them. If for some reason this goes spectacularly smoothly, I'll come back to the conservation. And by the way, the conservation is highly controversial what it all means, but let's not. So we're not going to go there though. But just as a reminder, this is the one. You kind of got three different folks yesterday giving you their somewhat different pictures, but all saying similar things that we can predict. Well, we have these characteristics. We have epigenetic features that are clearly characteristic of promoters, enhancers, insulators, and so forth. But we don't know at this point how important each of these features, whether there's particular histomodifications or presence or absence of a certain protein, how important are they for reliably predicting an enhancer? So if you pick so this is the part I'm going to skip over, so you can turn the pages as I skip over it, and get to this thing that's got, this picture of, this is Nergi's, if I could get to the right page, Nergi's Doan, who did, this is her thesis work with me. And even though you will see it and hear it many times that histone modifications are a good predictor of enhancers or promoters, if you've got the information on coactivators or really key transcription factors, they're actually better predictors. And Nergi's did a study that I think helps give us some insight into the contributions of each of these features. So I want to start with this, and then we'll go through a bunch of other assays that people have done. Okay, so the insight that Nergi started with, and were several papers from Gerd Blobel, and we were working with him on several of these and others, that had argued that in erythropoiesis, because there are many transcription factors that are needed, but a key one is GATA1, and sometimes is working in combination with a protein called tau1, which is a heterodimer with an E protein, E47. And multiple studies had said that combination of two transcription factors was, virtually every time you saw that, the target gene, if you could figure it out, was induced. So it's a really strong relationship. So, and we'd already used chip data against GATA1 and to ask, well, how good a predictor is this of being enhancers? And it worked pretty well, but it was kind of around this very common result of about, half of your predictions worked and about half of them didn't. And so I said, well, let's focus in on tau1. So she agreed to do that. Now, almost every place that we see tau1 bound in these maturing erythroblasts, and this is the signal for tau1. This is in a dense view. This is a nice strong signal within the a large intron of this gene for glycophorin C. There's you see tau1. You can also see GATA1. These are just different cell types. Some of these are cell line models. Some of these are primary cells. And there were these hypersensitive sites determined either in our lab or in John Stamatoyiannopoulos' lab. And then if we looked at the H3K4 monomethylation, we saw that is a dominant signal at many of these sites. All the things that David was talking with you about yesterday. So she incorporated two assays. This is the one that she ran herself, where she picked out 70 of these tau-1 bound sites, and did a transient transfection, which is that we looked at that reporter gene assay yesterday, and it was looking at the ability to drive luciferase in a cell line called K562. And she did it very thoroughly, you know, multiple biological replicates, multiple technical replicates, and then would summarize these as these box plots for the activity across all the determinations. And we set what I'm going to argue is a very conservative threshold for calling things as being enhancers at this twofold level. I mean, statistically, even things down in this level are significant. But we said, well, let's say we're going to make a call for enhancer. If it's above 2, we'll say it's inactive. If it's you know, below 1.5, and we just accepted there's this hard to call region. It's actually clarifying. If you got, you know, anytime you apply a threshold, you're going to have some results that are sitting right above the threshold, some that are right below it, and you know they're really not different, right? So, but you still need to make a call, and sometimes you can give yourself some intellectual freedom, if you will, to say, that's just hard to call. So we're not going to really incorporate that. We're going to actually, instead of doing a binary decision, we're going to have these 3 categories. But even with this conservative thing, 56% of them worked. And you could say there are any number of limitations to this assay. It is a funky cell line that we're putting it into. It does have some erythroid properties. Again, as I said yesterday, we're not looking at the endogenous locus. And I said, well, boy, it'd be really nice if we had additional information. So it turns out Pinocchio and his colleagues have been so busy assaying around many different DNA segments, some of them but because of conservation, some because of coactivator binding, that we were able to find an almost equal number of DNA segments that we could see were bound by tau-1 in our cell lines or in the erythroblasts, but he'd already tested them, and they and a lot of them were positive for midbrain or limb or forebrain, and the positives, it was like 65% of them had actually worked. So so maybe the context really has kind of changed a little bit here, in that we used to be doing these assays when enhancers were hard to predict. You know, it was nice to find some that worked. Now we're starting to think we know what enhancers look like. We think we know what they look like, and we've been talking about that all yesterday. So if we know what they look like, how come they aren't all behaving the same? And is it an assay limitation? Well, that's probably part of it, but 2 very different assays are giving us rather similar numbers. Let me see, yeah, so this kind of illustrates it. The things that don't pass our threshold, this is just a pile up of the CHIP-seq reads for this one region, and it is every bit as good a peak of tau-1 as this one that has a substantial boost in activity. What else is going on? Are there other motifs or whatever, or maybe there's some of these other features? Right now we just did the one, right? But we can start looking at other features and see if anything actually makes a difference. So she did that. This is now still just looking at the 70 DNA segments that she had tested. And of course, so now we went through, and she just filled in, okay, I'm sorry, this is first number. So there's 70 rows here, and the columns are, each row is one of those tested tau-1 bound, oh, wait, no, this isn't the 70. This is more. I'm sorry. I should have flipped it. We've been running this assay for quite a while, and she compiled everything that we had done. I apologize for the switch here. So she took the 70 that she did 60 from another guy, and X from another previous student, things that we had published. So now we've got a set of 270 DNA segments. I think that's the right number. And then asked well, is there a DNase hypersynthesis? Is it accessible to DNase? Is it bound by tau1? Is it bound by GATA1? Is it bound by P300? This is an orthogonal data set from mouse cells. And these histone modifications that David was talking with you about yesterday, K4 mono and trimethylation, acetylation. The cage tags, that's actually a look at these enhancer RNAs we started to talk about yesterday. And here's these repressive marks, right? So this is, if you will, the predictor variables, and this is how well it works. So she colored it red if it was in that active zone. The threshold, the part that's hard to call, is pink and blue down there. And first of all, you can see that a lot of these features go together. Right, so if you're bound by tau1, you're actually very likely going to have all these other features like so. You can get places where you've got some active histone marks, but without the histone modifications, and actually the validation rate really falls down from there. By the way, how you do this grouping is far from trivial. You can use k-means, which wasn't getting us very well. This was she applied some, oh God, what was it? DB scan, something like that. But it's a clustering method that's was designed to bring out the differentials more. Oh, and if you don't have any marks, so we did design negative controls, and you almost never, you can just almost never get a validation if you don't have any of those marks. Well, so that's kind of a little picture of it. Can we capture that in a more quantitative way? So we use this plotting convention as like a receiver operator characteristic curve. It's not really a curve because we're just, every data point is some group of features. HM means a histone modification, just the activating histone marks. Oh yeah, speaking of activating ones, you see you never have these repressive marks in the categories where we had a high validation rate. Anyway, so you can go through and, like here, this is everything that's in a hypersensitive site and asking, if you just take that one feature, how good is it at capturing what you want to find, the sensitivity? How good is it at rejecting what you don't want to find, the specificity? And it's quite sensitive, but the specificity is not so good. There are lots of ways to get about the same performance. So you can take P300, that's the P there, it's the single P, it's just, if that's all you looked at was P300, the co-activator, that's where you get. If all you did was look at tau1 in this cell type, where tau1 we know is important, it works pretty much like P300. Adding K27 acetylation to P300 really doesn't help very much. Oh, and maybe everybody's done these before, but I should say in this kind of ROC curve, noise will give you a line along here. If you've got a great discriminating model that you can really tell everything you want to see and reject everything you don't want to see, it goes right up to this 0-1 position. That's where you're trying to get to. And we've still got a long ways to go for that, but it doesn't look like adding more features is necessarily going to help a lot. And maybe what we're really getting back to is this issue of we need more words in our vocabulary to describe regulatory regions. And a key thing here is if you don't have, yeah.
How do you define an active enhancer? How do you define an active enhancer?
So this one is based on the transfection assay.
Oh, okay.
So that is it, and one, we just don't have as, well actually, so I think there are any number of people who are taking I think it's 2,000 transgenic mouse. So 2,000 fragments have been tested in the transgenic mice, right? You could do the same thing, right, for those. Hopefully they will come up with really informative models. I'm not sure, from what I've seen so far, that it's not so much better than what we got from these. But yeah, it would, I mean, any enhancer assay you could do. And we'll look at some more, these massively parallel ones. I wish they would go through, well, actually, you can see some of their figures that are kind of along these lines, trying to figure out what's really contributing to the both aspects of the accuracy. You got to get high sensitivity. High sensitivity with lousy specificity is a no-brainer, right? I mean, you can just you can let the genome be your predictor, right? There. Okay. Oh, and so I was going to point out down here. If you take some of these, look for places that have these active histone modifications, K4 monomethylation, for example, and none of these transcription factors, you get really lousy performance, right? So that's the. Now let me emphasize that there's places where you have the modification without the transcription factors. If you just again, the modifications around factor bound sites are working pretty well. So I think that so there's some good news here. Is that these really are strong predictors, but they're not absolute. You can't just look at the genomic data and know you've got active enhancers. And as we'll continue this discussion, what do we even mean by an active enhancer, right? And what else was I going to say here? Yeah, and I want to just reiterate there. When the epigenetics is telling you that something has a really strong signal, but you're not seeing an activity in a particular assay, should you really change your assay or do something else? Or is it doing something that you're just not thinking about? You can say that these the things that don't validate that this guy is a false positive, right? But it's a false positive. It's important to state operationally what it's a false positive for, because it still might be a true positive for something else. That's not a deeply insightful observation, but I thought I want to.
Terribly skeptical here.
Oh, goody! I finally got your riles.
Presumably, the data you're showing is like the best CRE-seq, and the curve. So in some sense, it's the best you can do at so if you had a worse antibody for your transcription factors, or imagine somehow there were a more specific antibody for his histone modifications or something like that, that would shift the curve. Right? So particularly if we thought we had a worse antibody that was either missing more things or binding places it wasn't supposed to for the transcription factor, that would make the RC curve work.
Yeah, and this may not be so much technical skills, although my technical skills are crappy, but I got good people, working with me, so they have good technical skills, and those are pretty clean data sets. You can come on in. We may have picked a pretty fortuitous stage to look at the tau1, because there are other stages when there are a lot more bound sites, and it could be that these are the ones that are staying on as the old erythroblast nucleus is starting to get condensed, and maybe there may already been a biological filtering, to get rid of some of these other guys. But there's also another thing. There's a thing. I'm glad I finally remembered something I meant to say because of your question. One of the things that did, well, I'm going to go back to the figure. We did search. Yeah, I took these slides out, but obviously, one would want to look at motifs in the bound segments that were active and the ones that were not. And I said, we have to do this because every reviewer is going to ask for it. I actually had no confidence that we would find it. I mean, I knew you would find some motifs, but it says, yeah, what do you, motifs, you can always find motifs, right? It turns out she found SMAD binding sites and some others. And then we found there were some other data sets where people, I wasn't thinking about SMADs because SMADs come on when you treat yourselves with BMP4, and we hadn't done that treatment. So to me, it was just a whole nother experiment. Well, it turns out the SMAD1 was, when it was under that stimulated condition, the SMAD1 was going preferentially to the sites that were active as enhancers. And Then I heard Shirley Liu talk about a study that she had done with her collaborator, this is published now, they'd done notch binding sites and tried to see which ones were active in some assay or not. And they had broken out into the stably bound notch sites and the ones that were dynamic. And the dynamic ones were enriched in the ones that were active. So I think maybe what's going on is we picked a nice window and a dynamically active binding protein. So when we did this with GATA1, the numbers weren't as good. And when people did it with MyoD, they got, they went to a higher, let's just say that they got about what we got with GATA1, right? So I think it's the dynamics that could be a real key here. So yeah, quality is always important, but working within a dynamic system might actually give you better predictors. Any other questions or discussion on that? So now, this is not what people would call integrative analysis. It just, pulls everything together. It's a visual integration. David started talking about integrative analyses yesterday, and let me just say we also were running CHROME-HMM. Again, that comes from Jason Ernst and Manolis Kellis. And just as a reminder, one reason that they developed CHROME-HMM was this feature that any given DNA segment is going to will frequently have more than one histone modification in it. And so you'd like to have, I think the word is a multivariate segmentation procedure, but you get this objective definition of chromatin states. So we didn't have so many histone modifications, but it was enough for our purposes. And each state is characterized by, if you will, a frequency of having, or if you're thinking in hidden Markov model terms, frequency of emitting a certain a signal, a certain modification from those states. And so we did that, this is that comparison between CHROME-HMM and Segway that David mentioned, and they do tend to find roughly similar states. This is by far, this is not a solved issue, what's the best way to do segmentations, but I won't get into that too much. The reason I'm mentioning that is this Markov modeling to define chromatin states was about the only product that phase 2 of ENCODE released to say this, and we actually never really said this, but we sort of was inferred, this is our best guess at regulatory regions. This is actually digging down into one of the figures from this 2012 paper, the segmentations had predicted that region as being an enhancer. And then actually, this is post hoc. I looked into to see if newer data also supported it, and it did. And this is one of the figures in that paper where that enhancer, when you did an assay, kind of like I was talking about for it's kind of a combination of the two assays I just talked about. It's not transgenic mice, it's transgenic fish, and your reporter is a green fluorescent protein. And this is actually you can then examine the Medaka fish while they're still alive, and you can actually see these green cells. These are erythrocytes that have turned green because of the GFP, and they're circulating around through there. So, but of course, in this paper, we never said what the failure rate was. And several people were wondering, how well does this really work? So the deal for the integration was to give you, just to remind you that it's the CHROME-HMM predictions that people then looked at in much larger scope instead of picking a handful to show you. Let's look at hundreds of them. So ramp up the scale of the assays, use massively parallel enhancer assays. And about 2-3 years ago, around 2012, multiple groups, Jay Shinjuri, Tarjay Mickelson, and Barak Cohen, all published methods along these lines, and I'm going to go use Barak's data, and well, anyway, but you could talk about similar things from others. So here's the innovation. So we need, instead of doing 70, which was a big deal for Nergis's project, or hundreds of them, a few hundred. Let's try to get up more towards 1,000 cis-regulatory modules. And so the idea is to use a sequencing-based assay for every predicted cis-regulatory. Well, I'm going to continue to say modules. Barak calls them CREs, cis-regulatory elements, because he's looking at smaller segments. But anyway, this is basically your predicted enhancer in this assay. And you've got a readout that's a red fluorescent protein. And the key thing is that for every construct where you've got a predicted regulatory region, you put a barcode. All right, and so now you can do thousands of predicted regulatory regions. In this case, you notice he's talking about variance. He actually, in this particular paper, this first one, he took a known regulatory region and he did saturation mutagenesis, changed every base to every other base. That was the context here. But it turns into a way to look at lots of different predictions. And he, this would not have been the starting point for the essay that I would have wanted to do, but I guess he was collaborating with someone who was interested in retinal gene regulation in retina, so they actually took mouse retina and electroporated the library into this. A lot easier just to zap your stuff into cells. But the point is, and then you can grow up a big population of cells that have been transfected by this library and sequence the RNA and sequence the DNA. And all of a sudden, you're only a step or two away from having thousands of measurements for the activity of each one of these. And you can make multiple versions of each of these variants, So the readout is, so expression level driven by this first variant is the number of tags you get, sequencing tags you get from the cDNA library after doing the electroporation in this case, divided by the number of, the tags are just the barcodes, right? So how many times do you see a particular barcode in the RNA? How many times do you see that barcode in the DNA? You just take that ratio, and voila, you have this nice result. He actually, this has to do with the red showing up in the right part of the retina, and this is comparing what he gets from his CRE-seq versus what you get in a traditional assay along the lines that we were just talking about, and it works very well. Okay, so now you can really ramp up this code. Any questions about that? So we're gonna hit several of these high throughput assays now. So well, by the next year, Barak's group did something that's a little closer to what the context we're talking about, which was how well is a particular transcription factor a predictor for enhancer activity. And this CBX is I guess a known factor. I think it's still in the mouse retina now. And back to the construct here. Oh yeah, and I need to say a few other things here. You got your reporter that'll turn your cells red. You got a minimal promoter, and you've got your cis-regulatory element, you've got your barcode. Now the thing, how do you get thousands of these, you might be wondering? Well, you make a deal with Agilent to make to synthesize. These are all synthesized. So the sequence of the cis-regulatory element and the barcode and whatever else is needed to insert these into the vector is all synthesized. And I forget the trick he did, but you synthesize the barcode contiguously with the CRE, but then they did something to put the expression cassette in between them. There's some cloning trick. And And then you could look at the CBX bound regions and that have the motif, don't have the motif, unbound regions and all of that. And you see now looking at really hefty numbers, right? Hundreds. Now, and we know how we can get this ratio of RNA reads over DNA reads, take the log 2 of it, that's all fine. Where do you draw the line? Now you can see where Barak drew the line, but where would you draw the line? And where you draw the line makes a huge difference, right, in how you do this. So the way he's doing it, and I think he's taking a very conservative approach, is he does the same, for everything he's going to test, he's also got a scrambled one. Remember, he's synthesizing the sequence, so you can do a scrambled version of it. And so he just empirically lets the scramble define your null distribution, and he's looking for anything in his positives that exceed that null distribution on the high end, say that's an enhancer. If it's lower, significantly lower, you can call that a silencer. Okay, any questions about that? All right, so that's, oh, and by the way, just like I was saying for tau1 in erythroid cells, this CBX is a good predictor of enhancers for this retinal assay. Now, back to the ENCODE data and the segmentations. So the chrome-HMM segmentations were interpreted as being indicative, or I better say predictive, of being strong enhancers, weak enhancers. Hold on, I got to remember what to say. Strong enhancers in K562, weak enhancers in K562, repressed in K562. That'd be, say, dominated by the polycomb mark, or the or looking at different a cell type that this is a human embryonic stem cell and predicted as an enhancer. And so what they did then was to synthesize a hefty sampling, hundreds of examples from the predicted strong enhancer class, K562, transfect that into K562 cells, the predicted weak enhancer class, the predicted repressed class, and then this is the predicted enhancers in stem cells. And let's see a little bit. So very good replication between assays. I mean, that's really tight for replicates across a large number of experiments, and a good correlation with between his assay and the regular transfection assay. And I got to try to remember here. This is, I think the fainter colors are the predicted regulatory regions, and the darker ones are the null distribution. And so an awful lot of the signals come out right here in the null distribution, but you do get a few coming out here as higher, and hence enhancers, and really not very many down lower than the null distribution. But the thing is, and that's really the pale green that's in there, I think, is that this thing, quote, weak enhancer predictors, they actually had the greatest number of positives. The things which the segmentation had predicted would be repressed, they were not active, and things predicted to be active only in stem cells were not active in K562. So I look at this as some good news and some challenging results. How well do we really understand what's going on? So let's go through this table, and because my memory's not so good, I actually included the notes here in case I probably will forget something. He actually did two different statistical tests, either the rank, Coxon rank order, or is that Pearson? Anyway, so compare the, of all the hundreds of candidate regulatory elements in each segmentation class, what fraction of those exceed the 95th percentile of the scrambled, and of course, in the brackets is the fraction of the scrambled that exceed that, and it really was the 95th percentile that was the cutoff. You can see that's all around 5%. And so this is what you get as active. And it seems to me the best you can say is that a quarter of the predictions are validated by this assay. And that's not very many. You know, so I think when this paper finally came out, the conclusion was stated delicately, right? But it's still, it's 25%, and the background's 5%, right? And you can do the statistical test differently. Okay, you can get up to maybe 39%. There's another paper from Manolis Kellis's group, Caridpour et al. It came out around the same time, and where it's very hard to get comparable numbers from the way they presented the data, right? But it's, I think that they were seeing something quite similar, although if Manolis were here, he would be much more positive about this. But I honestly, I don't see any reason why people had expected this to be a whole lot better. It's not horrible, you know, it does have some predictive power, but we still have a long ways to go. And so I think we, and that's why honestly, I don't see this as being necessarily bad news, but just saying there's a lot of room for improvement. And you don't always have the advantage of, like the study I started with that Nergis had done with me. We really knew that tau 1 was associated with an enhancement, and we had pretty nice data on it. And as I finally remember to point out, the fact that it's dynamically active makes a big difference. This is really just based on the panel of histone modifications, which have some predictive power, but I think it's good to boost it with some additional information. Okay. How about, any questions or discussions about that?
Because CBX5 is heterochromatin protein, and it should be they are correlated with heterochromatin. So in this case, I don't know if this CBX is another protein that might maybe correlate with active marks. So this is for me a little bit strange.
Well, I infer that you know a lot more about this than I do. It's been a long time since I read that paper with the CBX, and I was, and I didn't reread it recently. So I don't have any wherewithal to answer. But this is a really nice paper and it's well written. So please have a look at it. So I really don't know that protein myself at all. Sorry. No, but by the way, feel free to ask questions whether I know the answers or not. I'm not embarrassed. Okay, let's talk about some other assays. And there was someone was asking about STARR-seq after class, and that's a really nifty assay. And let me just remind myself of how I'm going to get through this. Okay, yeah. All right, so right. For the assay, the CRE-seq, you definitely, it's designed, it works by synthesizing the DNA segments you want. There's a limit to the length that you can synthesize, thousands of molecules, thousands of DNA molecules, and you may have noticed it, but the size that the Cohen lab was putting in was only about 84 base pairs long. Now, that's pretty short, and the enhancers that are really robustly active that I know of tend to be more of the order of 200 or so base pairs. And why are they large? Remember when I talk about 72 base pair repeat, it says multiple transcription factors binding. And genetic experiments, actually, for those of you who go down to the bar here at Cold Spring Harbor, you see the sign-up wind ships place, and I'm pretty sure that refers to Winship Her, who was the person who did those genetic knockouts of the 72 base pair repeats. And the experiment was this thing where he inactivated the enhancer, so he had this debilitated virus. He didn't get very many plaques by taking out, say, 2 out of 3 of those units. And then he selected for viruses that had rescued their ability to grow, that gave a lot of plaques, and he sequenced them, and what they had were now internal, that they had duplicated the bit of the remnant of the 72 base pair repeat that he had. So it was, the virus was saying, one's not enough, right? So anyway, that's a little local story. Anyway, so there's reason for concern that CRE-seq is not going to, it's going to miss some potentially active elements because you don't have enough. Wouldn't it be nice if we weren't constrained by the need to synthesize? And by the way, even if you get a good deal from Agilent, it's still expensive. And what we'd really like to do is to put in this case, the entire genome, genomic fragments, and what I'm much more interested in is taking a selected subset of the genome, and there are papers coming out about that. So this is from Alex Stark, the STARR-seq, and he actually designed this as a assay to find enhancers by activity. I think a lot of us are looking at it really as a way to say, well, of the things that I think might be enhancers, which ones are active? But he actually did, I think, a representative cloning of the whole Drosophila genome. And so let's look at the construct here. It has some things similar to what was mentioned before. You've got a reporter gene, and you're going to clone in DNA fragments. And some of these DNA fragments will be enhancers, and you'll get these RNAs being made. So basically, you just and you in this case you rig up then you've got a little a tag in on with your with the DNA segments that you're cloning in, so that you can just do the transfection and read out do the RNA-seq and look for the presence of the candidate enhancers that you had put in. Again, just doing a read counting assay to ask are each of the fragments that you put in actually enhancers, and this was his a comparison with the luciferase output, and you can see there's a very nice correlation there as well. The nifty thing about the STARR-seq is that you don't have that size limitation. You can clone in DNA segments, whether it's from just randomly sheared genomic DNA, which is what Stark did here, and he could select a certain threshold and say, here's a whole collection of enhancers, or you could do a capture approach, where you pull out a bunch of candidate regulatory regions, and capture STARR-seq was, I think, published earlier this year. I'm sort of like one step removed from this result, but as part of the mouse ENCODE project, a guy named Feng Yue had did this assay to look at, in this case, I think they were candidate enhancers predicted by the histone modification patterns in, I think it was embryonic stem cells, and made the library and did the assay, and looking at this sequence readout, the candidate enhancers, in this case, are really separated very nicely from the random controls. So that the STARR-seq is another way to look at a lot of candidate regulatory regions at once. Now, so how are we doing here for time? We got 10 o'clock. Let's go to how do we go beyond the single features or histone modifications only, and some of these chromHMM kind of segmentations, and get to something as a better predictor? Well, in principle, if you have enough training data, you should be able to do machine learning to find good classifiers. And a very pretty example of that is from Katie Pollard's group, came out last year in PLOS Computational Biology. And there are a few things I wanted to point out about this. First of all, Katie was well aware that motifs are associated with regulatory regions. She's well aware that constraint was a major contributor to regulatory regions, as we were discussing yesterday. And there are multiple epigenetic marks, different histone modifications or whatever, that could contribute. And if you have a sufficiently large number of DNA segments that have been tested, some of which don't work and some of which do work, you should be able to figure out a quantitative contribution of each one of these features to some kind of a function that's a discriminatory function. And so that's just a basic idea. I'm sure in execution it's not trivial, right? But that's the basic idea. And I think this was the tenfold cross-validation of what she came up with, and had a very nice ROC. And that ended up with a classifier that would make a prediction of it is likely to be an enhancer or not. And Katie will emphasize, she would actually probably be a little concerned with me right now because I didn't emphasize strongly enough the type of enhancer. She's predicting the types of enhancers that she trained on, and that is the enhancers that are active in specific tissues or groups of tissues at day 11.5 of gestation of the mouse. Doesn't say anything, doesn't necessarily tell you anything about adult brain or adult kidney or whatever, although it's not a bad thing to run anyway. And she has made predictions, I think both through human and mouse with these. And another thing I liked about this, because I think it's important to separate out these two issues, is come up with an algorithm that does a good job of separating active enhancers from DNA segments that are not active. And then she does a second round of training to try to get tissue specificity. So she separates out those two issues, which again, I think is a smart thing to do. And that these ROC curves are impressive, right? Again, what you want to do is to get a deflection up into this upper left-hand corner. And I think she's looking at here different inputs to her training were performing slightly differently, and more data was better. And I'm trying to remember over here. The whole composite of the functional genomics had a greater area under the curve that is more of a deflection to the left than some of these individual marks. And even though I've been raving about P300 in this particular assay, that didn't work as well as having in all these other marks. And this is just an example of having the predictions all across the locus, and some of these were already known to be active, and this was one that had not been previously looked at, and it is active. So machine learning is is a very good approach, right? And you can get strong discrimination. Getting more data sets, more large data sets to do the training on would be, it would be a big help. And I look forward to the time when our predictions are getting well beyond the, say, 2/3 success rate, which various studies are kind of hitting right at that. But maybe that's the best we're going to get. We'll have to see. Any questions on that? All right, let me remind myself of where I'm going with it. Oh, yeah, we need to get to this quickly. Okay, all right. So the STARR-seq, Alex developed that, as I said before, to actually select by, I don't know if I should have emphasized this more, but that initial CRE-seq assay, the readout is just from the sequencing the tags in the RNA and the DNA. In this case, I think you're actually using the fluorescent protein as a readout to select for the things that are active and then sequencing those. So STARR-seq was the first one of these assays directly for function. There are several others that have come out, and I'll just point you to this this issue of Nature Methods, where in one case they were using an approach of just letting the nucleases, in this case restriction enzymes, cut at accessible sites, basically using this nuclease accessibility as an assay to liberate likely regulatory regions, and then inserting them upstream of a GFP, transduce this in and select for the green cells. And there are other variations on that. That one from Pinocchio's lab. This is called Firewalk from those assays. There's a large sector of geneticists who don't like these reporter gene gain of function assays. I'm not one of them. Actually, I'm not a geneticist either, but anyway, these and all those things I've been kind of saying, oh, well, of course there are limitations to the assay, yada, yada, yada. Well, yada, yada, yada not the way they react. They say that means that the fact that it's not integrated in the right place and the chromosome means that your results are completely irrelevant. And I argue back, but you know, I've yet to change anybody's mind. I have stood my ground, right? But the truth is that you want to complement these gain of function assays with loss of function assays. You may ask, well, why don't you just go there anyway? And it's because an awful lot of time, loss of function experiments in regulatory regions give you no phenotype. And it used to be extremely expensive to do it. So you do extremely expensive, long time, get no phenotype. It's depressing. Now it's easy to do it. So, you know, the fact that a lot of these, I still bet a lot of these aren't going to generate a phenotype, but at least we'll be getting a lot more experiments done. So everybody's much more excited about loss of function. And it is, again, you can thank whoever you want to thank for CRISPR. You can thank them for this. And I want to talk a few minutes. Let me just show you some of the stuff that's happening right now in this. And I need to explain a slightly complicated system so that I can talk about this paper that just came out last month. All right, so numbers, we're staying in red cells, and of course red cells are full of hemoglobin, and the hemoglobin, there's an alpha globins from one locus, beta globins from another. And there's several of these globin genes on the beta globin genes. And they're expressed at different times of development. So you've got an embryonic form here. This is, in humans, you and I express these gamma globins to make a fetal hemoglobin. And now all of us are making either the wild type hemoglobin A or it's probably unlikely. There might be some people heterozygous, but a common variant is the sickle cell allele, HBBS, and that's expressed in adult life. If you're homozygous for this variant of hemoglobin A, the hemoglobin S, of course you have a sickle cell disease, and in most genetic backgrounds, this can be a really debilitating disease. It's a huge health burden in many populations. So there's a lot of interest in trying to improve the life for folks with sickle cell disease. And we've known for a long time that, you know, all of us express these gamma globin genes in fetal life. We all differ in the amount of hemoglobin F we continue to make in adult life. This is a quantitative trait. It's genetically determined. There are multiple loci that determine this. And if you have, but the interest is, if you have a fairly high, instead of having this low level of expression, there are people who have moderately high levels of hemoglobin F, and some people have moderately high hemoglobin F and sickle cell, and they have much milder symptoms. This has been known for decades, right? So the milder symptoms you get with hemoglobin F. So there's been a huge amount of interest in how do we, if you will, reactivate or the gamma globin genes or keep them on in a context of sickle cell disease or other inherited anemias like thalassemia. Okay, so, sorry if that's kind of a complicated story, but you have to know that background to understand why people are so interested in this. Okay, so whether you have low levels of hemoglobin F as an adult or modest levels is a quantitative trait, and it's inherited, and the genetic determinants of those of that variation in hemoglobin F map to the globin gene locus itself, including some of these cis-regulatory modules, and to 3 other loci: BCL11A, KLF1, and MIB. BCL11A has gotten an awful lot of attention. And it turns out, and this is through work from Dan Bauer and Stu Orkin and multiple people in his group and other groups, this BCL11A is a repressor. And it's expressed in lots of different cell types, brain and lymphoid cells, but it's also expressed in erythroid cells. And when it's expressed in erythroid cells in the adult, it serves as a repressor of gamma globin. And it turns out that some of these naturally occurring variants, the things that the people mapped by GWAS to say this locus is involved in regulating gamma globin, they map to an intronic enhancer, and this is drawn as 3 diamonds here because there's 3 hypersensitive sites. All right, so, and there's been, and then this is like really amazing, and I hope it continues to be true as more data come in, but current knowledge is now telling us this is a exclusively erythroid enhancer. So we have this ubiquitous, almost ubiquitously expressed protein, and the B and C BCL, I think, stands for B-cell leukemia. You know, if you mess around with these guys, you know, you can have problems. But if we could knock out that activity just in erythroid cells, we'd lose the repressor that's keeping the gamma globin genes off, and we could force higher hemoglobin F. That's the dream, right? And for years, I'd say, this is crazy, right? I mean, you can't mess around with people's, even somatics, you know, you're gonna mess around with an essential enhancer, but the erythroid specificity makes this possible. It's kind of like, you know, nature's already set up the GAL4 system that you want for tissue-specific targeting. So that's the dream. Can you actually force a deletion and get even more improvement? And of course, with CRISPR, you can design knockouts pretty well. So the latest version of this story, it just was released online last month, is from Nature. It's again from the Bauer lab and Stu and one of the CRISPR originators said, let's find the Achilles, if you will. The metaphor is the Achilles heel. This is like 3 hypersensitive sites over about 15 kb or so. And yeah, you could think about completely knocking it out in using 2 guide RNAs for your CRISPR and all that. But boy, what if maybe there's a really sensitive binding site, some motif that you could really hit and knock it out. So that was the motivation here for this beautiful paper that just, again, is coming out. And they actually call it saturation mutagenesis. And it's close, it's pretty high density. Now, I'm no expert on CRISPR. Many of you probably know more about it than I do, but just as a reminder, one of the wonderful things about it is you can get a single guide RNA to direct Cas9 to do a cleavage at a place that matches that sequence, but you do have to connect it in with this PAM, this 3 nucleotide sequence, right? So that does limit the density that you can design short or single guide RNAs to do this mutagenesis. But you can come up with hundreds of these single guide RNAs, put them into an appropriate transducing vector, and transduce them in. And now, in order to make this work, here's another key thing. You need a phenotype you can look at, right? So what that they did, they were able to actually monitor changes in hemoglobin F. There's the cell line HUDEP cells that where you can really see this come up. So then you can pull out the cells where you've actually shifted the level of hemoglobin F by doing this mutagenesis in BCL11A, and then then sequence, to see which short guide RNAs actually had worked and where the cleavage sites were and so forth. And you can get these scans, right? So this is looking across one of those hypersensitive sites, the one at plus 58. Oh yeah, that's the hypersensitive site. This is actually one of those chrome-HMM, looks like a binary state thing. But anyway, so you can look, these are each of the places that were hit by Cas9. Cas9 hits, so there's a little error-prone or deletional repair that goes on, and you get these mutations, and you get these spikes in activity. So I mean, this is this is really pretty exciting. Obviously, it's a loss of function assay, and let me emphasize the great thing about that is you're going right into the genomic context that you want to look at. And the surprise was, where's the, in here, let me get this right. Okay, so the greater the deflection, the higher it is on the y-axis, these points, that the greater in effect the targeting RNA had. And you see this peaking around in this zone is very high resolution here. It's only 10 base pairs apart. And the places being hit are not in, the places that give the greatest response are not actually in the well-known motifs. They're a few base pairs away, right? So it's nice to, so if I were doing the predictions, I'd say, okay, I know the GATA1 binding site, I know the binding sites. Everybody knows the binding sites for all of these because you can look them up in the databases. And that gets you close, but not exactly into the hot point. And yeah, so the hottest one is close to things that are conserved and have a motif. I mean, like a few base pairs away. Well, I shouldn't belabor this too much, but the reason I wanted to at least get to here, and I think this is probably a good stopping point, is I decided, well, let's just start talking about a bunch of different assays and what they might mean, but we finally are at the point where we can start doing a whole battery of assays. I mean, I started out the lecture saying, well, wasn't it great that we could do both transient transfections into a cell line that's been informative and look at a similar set of fragments in transgenic mice, but they're all gain-of-function assays. And it's perfectly reasonable to go in and, for one candidate enhancer at a time, go in and knock it out. You need two guide RNAs to do that. Or, people are already ramped it up to hitting almost everything. Let the readout, the phenotypic readout, guide you to the places that are most sensitive to perturbation and presumably most important for that enhancer activity. And I would also, I guess, so in summary, I'm saying I love all these assays. Some of them cost a lot more than others, but I would, for those of you who are going to be tasked with harvesting the insights from all this epigenetic information and do something meaningful with it, don't hesitate to do multiple orthogonal assays. Think hard about what they all mean. And that's it for me.